So in summary of the path to alpha chain rearrangement, we have our double positive thymocyte, which is expressing both CD4 and CD8, at which it will start to increase its RAG expression, which is necessary to catalyze the VDJ recombination. And the recombination event will proceed at one allele first, and so it will try and join the V to the J at one particular allele. And if this is successful at this particular allele, then we will have things like allelic exclusion. If it is unsuccessful, then we will move to the next allele to try and undergo rearrangement from joining the V to the J of the other alpha allele. And if this is successful, once again, the cell will be able to move on to the next cellular stage. And if this is unsuccessful, then this will result in cell death because no alpha chain was produced, and so it cannot produce a T cell receptor that will allow it to move on. So then, looking at the particular T cells that have had successful rearrangement of their alpha chain, these will move from the double positive stage to the single positive stage. So if you remember us talking about mature T cells, they're only going to express one co-receptor, either CD4 or CD8. And this is particularly important for the effector function in which they will take on during the immune response. And so before we can have a mature T cell, we need to have it such that the cell is only expressing one co-receptor. So now we're going to talk about the process by which only one co-receptor is expressed. Moving to the single positive stage. So here it's important to remember, which is something that we've talked about previously in our antigen presentation and lymphocyte antigen receptor lectures, is that T cells are MHC restricted, meaning that T cells will only recognize and interact with their antigen when it is presented as a peptide form within either MHC class 1 or MHC class 2. And this differs from antigen receptor recognition in B cells because antibodies do not require presentation through these MHCs, and so they are able to interact with native antigens that are folded. However, T cell receptors must bind to their peptide through either MHC class 2 in the case of a CD4 T cell, or MHC class 1 in the case of a CD8 T cell. And so this is where these co-receptors are important because they will facilitate interactions with MHC that are antigen independent. So you can see these interactions with the MHC are occurring on the outside of the MHC molecule and not facilitating the interaction with the antigen that is shown here. And so this is just pointing that interaction out. The process by which we move from having two co-receptors to one co-receptor is referred to as positive selection. So in the event, in the thymus, that this double positive cell is able to interact with its TCR through MHC class 1, then this will trigger a response in the cell to start only expressing CD8 instead of expressing both CD8 and CD4. Conversely, if we have a T cell receptor that is able to interact with MHC class 2, and then the T cell receptor will, because of this interaction, it will tell the T cell to start expressing only CD4 and not to express CD8. So this is how you can distinguish between what type of alpha beta T cell you will become and how we move from the double positive stage to the single positive stage. And something that's important to keep in mind here is that while allelic exclusion is very stringent, particularly with B cells and their light chains, it is not the case during alpha chain rearrangement. This alpha chain rearrangement will continue to happen until positive selection has happened. So not until the interaction with the thymic stroma, which allows for commitment towards one particular co-receptor occurs, do we have the initiation to stop alpha chain rearrangement. And so this is just one of the ways in which T cell development differs from B cell development. And so it's not until this particular stage in which there's the interaction with either MHC class 1 or MHC class 2, at which point we can turn off our alpha chain rearrangement. Now, this may seem like this could be a problem for the particular T cell because, as mentioned extensively in the B cell receptor, or in the B cell development lecture, this allelic exclusion is necessary to ensure that there's only one B cell receptor for every one B cell. So you may be asking yourself, how does this not result in two T cell receptors potentially within a single T cell? However, the reason that positive selection is important here is that, first of all, it's unlikely that there would be a functional receptor produced off of both alpha alleles, just because of the way that recombination occurs. Secondly, it is very unlikely that if there were two different T cell receptors that had two different alpha chains, that they would both be able to interact with the same MHC and both require the same co-receptor. So for that reason, it's not really a risk to the T cell to have two T cell receptors just because alpha allelic exclusion is not as stringent as it is with B cell receptors. And this is exclusive to T cells because they're MHC restricted. So at this point, we are at the single positive stage, but the T cells need to undergo one final stage before they are ready to mature, and this involves the elimination of autoreactive T cells. And this is a process referred to as negative selection, and it differs a little bit from the negative selection that we learned about in B cell development, namely that it's much more stringent. So this can happen either within the cortex or the medulla, where if there is an interaction with a T cell receptor with a particular self-antigen, then this results in killing of the thymocyte. And so unlike B cell receptor negative selection, in which the B cell can have different fates such as anergy or receptor editing, at this particular stage in T cell development, negative selection is very stringent. And so this is the site of positive selection. The reason I want to point this out is this elimination of self-reactive T cells can happen during positive selection, which occurs within the cortex of the thymus. But this can also happen by other cells that are present within the medulla of the thymus. And so at this point, again, if the T cell receptor interacts with self-antigen, it will be eliminated and killed by apoptosis. And so there's particular cells within the medulla region specifically, and these cells are either macrophages that are bone marrow derived, or these specialized cells within the thymus referred to as medullary thymic epithelial cells, which is often abbreviated as MTECs. And these will present antigen to the T cells as well. So this in particular, because it is in the medulla, is after positive selection. So just to keep in mind that this elimination of self-reactive T cells can happen either either concurrently or after positive selection. But there is a particular issue in which the thymus is only expressing thymus-specific antigens, but we want our T cells to not be reactive to any particular organs of the body that exist elsewhere. We don't want any T cells to escape the thymus that are potentially self-reactive. And the reason that T cell negative selection is a lot more stringent than B cell negative selection is that B cells are often dependent on T cells to elicit their immune response, whereas T cells can elicit strong immune responses that can lead to autoimmunity much more easily. And so thinking back to these medullary thymic epithelial cells, these MTEC cells within the thymus, they have this special transcription factor that's referred to as AIR, and This allows for expression of antigens that would be present in other organs. For example, expression of insulin, which is present in the thymus, as a way to select against T cells within the thymus that could be self-reacting against insulin, leading to autoimmune diseases such as type 1 diabetes. So at this particular stage, these medullary thymic epithelial cells, as their name suggests, which are located in the medulla, they can interact with these T cells, and if the T cell receptor on these T cells interacts with these MTECs, then it allows the cells to undergo apoptosis so that we are not having a release of these cells that can lead to autoimmunity. So this is another way in which we can have very stringent selection that is not only selecting against antigens that are present in the thymus, but also antigens that are present in other organs as well. So just to give a summary, there is this particular model called the Affinity Model that can explain the ideal interaction of a TCR with its MHC peptide in order to undergo proper development. So in this particular part of the graph, these are T cell receptors that have no affinity for MHC. So they cannot undergo positive selection because they don't interact with MHC1 or MHC2 at all. And so they are referred to as often undergoing death by neglect because they're not receiving the necessary signals in which, when they interact with these thymic epithelial cells during positive selection, they get these survival signals, and so they are not able to undergo this particular interaction. Conversely, if there are cells that are T cell receptors that are interacting too strongly with the particular MHC peptide in the thymus, either in the cortical region or the medullary region, these T cell receptors will be eliminated by negative selection because they could lead to autoimmunity. So you want to eliminate them so that they cannot escape the thymus and lead to autoimmunity. So ideally, we would have an interaction of the T cell receptor that is somewhere in between, something that can interact with either MHC1 or MHC2, but such that the interaction is not so strong that it leads to elimination because of a potential chance to cause autoimmunity. I also want to point out this little sliver right here. There is a chance in which a T cell that is interacting fairly strongly with self-antigen can actually be converted into these particular types of T cells referred to as regulatory T cells. And so at this point in which they're interacting with self-antigen, they're converted into these T cells that are able to release suppressor signals when they see their self-antigen in the periphery. And you'll learn more about this in the autoimmunity lecture, but this is very important for these particular T cells, T regulatory cells, to exist because they suppress immune responses of other cells, such as B cells in the periphery, when they try and respond to self-antigens. And so then the last step of T cell development following this stringent negative selection is actually exiting the thymus and moving into the periphery and the secondary lymphoid organs. So much like T cell development at this point, much like B cell development, these are T cells, much like B cell development, these T cells will upregulate certain signals like S1P receptor that allow them to enter into the bloodstream. If you remember from our B cell development lecture, there's a gradient such that there's a very high amount of S1P in the blood and a very low amount in the thymus and the bone marrow. So this encourages the T cells to egress or migrate out of the thymus into the blood. There are also other receptors that are developed on the T cell at this particular moment, such as CD62L, and this allows for homing of T cells to specific tissues, just another encourager of where it should be located post-maturation. So, as I had mentioned before, I just wanted to review how thymocytes will actually migrate through the thymus as they're developing. So if you remember from before, they enter the thymus through these venules where they came from the bone marrow, traveled through the circulation, and enter these venules and start committing to the T cell lineage. And this is in the medullary region of the thymus. Then they will move to the cortical region, and this is where receptor rearrangement occurs and positive selection occurs. And then they will move to the medullary region for the last stages of development, such as maturation and egress. And as mentioned previously, the negative selection can happen either in the medulla or the cortex. So it's not necessarily dependent on where the T cell is in the thymus at that point in time. Okay, so at this point, I just want to give a summary of T cell development. So we start out with our progenitor cells in the bone marrow, and these will migrate through the periphery into the thymus, at which point they start this double negative stage. They will start to upregulate their RAG expression in preparation for VDJ recombination, at which they will start to undergo rearrangement of their beta or their gamma chain. We didn't talk specifically in depth about the gamma delta T cell receptors, but this would happen concurrently with beta chain rearrangement. And then at this point, we would have our double positive cell stage in which the cells start to express the pre-T cell receptor alpha chain to test for the proper rearrangement of the beta chain. And the gamma delta T cells at this particular point in time do not need to undergo the same selection processes as the alpha beta T cells. So if they commit to the gamma delta T cell lineage, this is the point at which they would be exported into the periphery. So getting back to our double positive T cells, they're receiving signals through their pre-T cell receptor if they've undergone proper beta chain rearrangement, at which alpha chain rearrangement will occur following the double positive stage, or following this stage, and then it will allow the cells to express both an alpha and a beta chain. This is the time at which positive selection will occur. And after positive selection, the T cells will either be able to interact with MHC class 1, at which they will become the CD8 positive, single positive T cells, or they will interact with MHC class 2 and they will become the CD4 positive, single positive T cells. And negative selection can either happen concurrently with positive selection, or it will happen at the single positive stage, but it is necessary in order for the T cells to fully develop and move into the periphery. Now that we have reviewed T cell development, just as a refresher, I want to compare and contrast B cell development and T cell development because, as you probably appreciate, there are many similarities, but there also are key differences. So the B cell receptor will begin rearrangement of its heavy chain, which is very similar to the T cell receptor undergoing rearrangement of its beta chain. And both of these will start out with the D to J rearrangement and then move from the V to DJ rearrangement. So at this point, very similar. And then you have both the pre-T cell receptor, which has PT alpha, and the pre-B cell receptor, which is the surrogate light chain, participating in telling the cells whether or not proper beta chain rearrangement has happened in the case of T cells or proper heavy chain rearrangement has happened in the case of B cells. Both of these cell types will undergo chain pairing diversity because of the proliferation stimulated by either the pre-B cell receptor or the pre-T cell receptor. So at this point, still very similar, and both will have allelic exclusion at the heavy chain or at the beta chain, which are very analogous to each other. Then we have BCR rearrangement at the light chain, beginning with the kappa chain and then moving to the lambda chain. So this is one of the first divergences in T cell receptor development and B cell receptor development, in that there are 4 BCR allele light chains that can undergo rearrangement, as opposed to the T cell, which only has one type of light chain, this TCR alpha, so there are only 2 alleles in which recombination are possible. Another divergence, it happens at the stage of allelic exclusion. So if we remember, the B cell receptor, after forming a proper light chain, will enforce allelic exclusion of the other loci that have not undergone recombination, as a way to enforce only a single B cell receptor for every B cell. However, at this particular stage in T cells, there's no specific allelic exclusion of the alpha chain until after positive selection. Then another divergence between the 2 developments is that B cells do not undergo a formal type of positive selection, whereas T cells, it's necessary for their development to move from the single positive stage to move from the double positive stage to the single positive stage, so that they're only expressing one co-receptor. And the main reason for this difference between B cells and T cells is that T cell development and T cell receptors are MHC restricted, whereas antibodies and B cell receptors are not. And then next, there is a less stringent negative selection process that happens at B cell receptors, but they still do have a negative selection process. It just allows them to potentially undergo receptor editing, or they can move into this anergic state in which they'll often be eliminated in the periphery, or they can undergo cell death. And by contrast, T cell development is extremely stringent. So if there's a T cell receptor that interacts with a self-antigen in the thymus, whether it's in the cortex or the medulla, at this point, the T cell will be eliminated to prevent autoimmunity. Another big difference between B cell development and T cell development that we have not yet covered is that T cells and the thymus, actually, the thymic tissue will start to degrade over time and be replaced by adipose tissue. And as you might imagine, this leads to a lesser capacity for T cells to actually be developed in the thymus. So the bone marrow will continue to develop B cells over the course of a human's entire lifespan, constantly pumping out B cells as well as other blood cells. But the thymus, by contrast, will start to have a lesser capacity to make T cells over time. Now, you may be wondering, does this lead to us not having T cells later in life? But it does not to worry. The T cells at this point are longer lived. So one of the key contrasts between T cell development and B cell development and maturation is that T cells in circulation are much longer lived than B cells in circulation. So even though you produce most of your T cells early on in life, they will continue to live on with you, whereas B cells are short-lived and they're constantly produced throughout your entire life. So this